Hello everyone. Today I am going to go over a very important but complex topic of cell division, and that would be mitosis and meiosis. And so mitosis can be seen in all of our regular cells. Like if you get a scratch or something, our cells will heal and divide and make new cells to replace the old cells. Meiosis is seen in gamete cells, that would be sperm cells and egg cells, and we're going to go through each of the phases and highlight some of the key differences between the two. So we'll start with mitosis. First, we have our regular intermission here, where we have our chromosomes. Then we go into what is called prophase. In prophase, the chromosomes will duplicate. So now you have two sets of sister chromatids. And that is one of the key things that happens during prophase. Next, you have metaphase. In metaphase, the spindles and the duplicate chromosomes will line up along the axis like this, and these spindles form. Then they enter what's called anaphase. Now in anaphase, the cell gets ready to divide, and we see this septum begin to form, and the spindle fibers will pull apart the chromosomes. So you have one on each side. And finally, we have telophase. And telophase is the end. The two new cells are separated with their own chromosomes. They are an exact replica of the mother cell. Got your blue and your red. 2N, 2N, 2N. And so you get two diploid. That will be this right here, diploid. Two sets of chromosomes. Daughter cells. Now, let's take a look at meiosis. Now, like I said, meiosis are found in gametes. That's strictly reproductive. And so you start out with your regular chromosomes, regular cell, and then we enter what's called prophase 1. So in prophase 1, it's the same as before. Your cells will duplicate their chromosomes and get ready for cell division. Just like that. Now you have two sets of homologous sister chromatids. Then, during metaphase 1, this is a unique feature for meiosis that you don't see in mitosis. It's called homologous recombination. And in homologous recombination, these sister chromatids, they get together. So we have those ones there, and these ones here. They get together and cross over. They exchange genetic information. So it's gonna look like this, where you have on the end of this chromosome a piece of the other chromosome, and on the end of this chromosome you're going to have a piece of the other chromosome, and the same for these two here. And so they stay together, and then during metaphase 1, still in metaphase 1, the spindles form and connect with these guys. Now, instead of pulling apart sister chromatids that we see in mitosis, we're actually pulling apart the homologous chromosomes. So we still have two sets of chromosomes stuck together in anaphase 1 here. Still together in a set here. And then we have telophase 1. Where we finally have two diploid daughter cells, but it's not really done here. Because now we have to separate these guys. I don't remember which one was on which side. That's okay. So then we go through prophase 2 and metaphase 2. And so metaphase 2, this is where it becomes similar to mitosis. Spindle fibers form and our chromosomes line up on the axis. These ones too. And the spindles line up right down the middle. Then we enter the next phase, anaphase 2. I am bad with numbers today. Sorry, that's a blob. It's supposed to be the septum begins to form. just like mentioned before. And just like before, we'll have these sister chromatids. The homologous chromatids are pulled apart from each other. And we are almost done. Then we have telophase 2. This is the end where the cells are finally separated. And what's cool about this is we have 4 unique daughter cells. Let me double check to make sure I'm doing this right. Alright, so we have 4 unique daughter cells because of that homologous end joining that happened in prophase, metaphase 1. So we have 4 haploid because they have half the number of chromosomes we started with. Take a look here. We started with 4 chromosomes, and we're ending with 2 chromosomes. So that would be 1N. So we have 4 haploid daughter cells. And so those are the key differences between mitosis and meiosis. At the end of meiosis, we get 4 haploid daughter cells. And the end of mitosis, we get 2 diploid daughter cells. Now, knowing the difference between this is really important, especially if you want to take a genetics class later, or if you want to do work in a lab and work with things like CRISPR. It's really important to know the difference between these. Thank you for watching.